All right. Good morning, everyone. Welcome to today's MPG primer. Mandan Arbab is the Loddish Family Assistant Professor of Neurology at the Rosamund Stone Zander Translational Neuroscience Center at Boston Children's Hospital. During her PhD at the Hubrecht Institute in the Netherlands, she worked on iPSC modeling of neurological diseases and CRISPR technologies. She completed her postdoctoral training with David Liu at the Broad Institute, where she published papers on high-throughput characterization of CRISPR tools and developed an in vivo genome editing therapeutics for neurological diseases. Her lab, which she started in 2023, continues to pursue the development of genome editing therapeutics for various genetic conditions and works to translate these drugs from bench to bedside. And today she's going to be talking about a recent paper called Base Editing Triplet Repeats of Huntington's Disease and Friedrich's Ataxia. Thank you.
Very much for that introduction. Okay. Yes, that's exactly what I'm here to speak about today, so I'm going to jump right in. Our lab focuses on the development of genome editing therapeutics for various different neurological diseases, starting from identification of good targets and strategies therefore, and then first validating these in vitro, and then ultimately taking them into animal models with the end goal of taking them into clinic. And so also doing the IND enabling studies for that. And we're mostly been focused on neurological disorders that affect movement, including motor neuron diseases, as well as repeat expansion disorders, which I'm mainly going to be focusing on today. So I'm confident that you are all well aware of CRISPR and base editing, so it obviously doesn't need much introduction, but I just briefly want to highlight a few aspects of these tools that are going to be relevant that you maybe don't think about as much. So base editing is a technology that utilizes the sort of search and find function of Cas9, but couples the Cas9 protein to a deaminase enzyme. And what this allows is that when Cas9 binds and the guide RNA hybridizes with one of the strands of the DNA, the other single-stranded portion of DNA becomes available for this chemistry by the deaminase enzyme to be able to happen. And in the case of, for example, an adenine base editor, it can turn this adenosine into an inosine that is recognized as a G. And then through DNA repair, this mismatched position here is resolved into a stable base, ultimately turning an AT pair into a GC pair. So in principle, I guess the two things I want to highlight, any single-stranded DNA is made available to the deaminase. Obviously, this is localized by the Cas9, but this is also what stops the deaminase from, in general, just deaminating all of the genome. And also, base editors typically will use not a dead Cas9 that only searches and finds, but in fact a nickase. And the role of the nickase is to nick the strand that opposes the one where the deamination event occurs. And the reason for this is that nicks are more rapidly identified by DNA repair as being problematic. And so you will typically then end up having the strand that had the original nucleotide of the pair on it being resected and templated off of the strand that has the deaminated position. And so you are in fact biasing then your outcomes to resolving in favor of the change that you've made rather than reverting 50-50 or less than that to the original base pair that was there. So initially, a couple of years ago, we demonstrated the use of this adenine base editing technology to treat a different disorder. This is a motor neuron disease called spinal muscular atrophy, or SMA. In this disorder, most often infants, pretty much as soon as they are born, begin to present with symptoms, ultimately leading to paralysis. And if untreated, these patients will not survive past about 18 months. And SMA is, in fact, the leading genetic cause of infant death globally. And even though to this day, we still don't actually understand the mechanism of pathology that leads to the loss of motor neurons, we've known for about 3 decades that this disease is caused by the loss of this essential SMN1 gene with retention always of this SMN2 gene, which is almost identical to SMN1 except for one C to T transition that causes a splice variant. And as a result, SMN2 genes produce only about 10% of the functional protein that SMN1 would. And the different subclasses shown here of severity actually relate to the copy number of SMN2 that patients have. So it was known that it's a dose-responsive gene. If you up the amount of SMN, you can also restore this disorder. So we developed a base editing strategy to treat this, shown here. So we turn this AT pair into the GC pair that is the one that is found typically in SMN1 genes. And after validating this in vitro, we delivered this into animals using AV9. AV9, importantly, is a vector that is used to deliver SMN gene therapy to SMA patients very successfully in the clinic. So we have prior knowledge, not only in animal models, but in patients, that this actually does deliver cargo efficaciously to the target tissue of interest. And what we were happy to find is that in neurons that get transduced in vivo, about 90% of them have this conversion of the insufficient SMN2 gene to the SMN1 gene. And through some additional optimizations, so the mouse model that we were working with for SMA is one that has a very rapid decline. So do the human patients, but in animals, what this means is that if you don't rescue the SMN levels by about 5 days after birth, you really aren't rescuing their survival at all. Their median survival is about 17 days without any treatment. What we did is we used a bridging drug, essentially, one of the transient therapeutics that's on the market for SMA that can very briefly, by rescuing splicing, ameliorate disease. So we gave a very small dose, only a single time, at time point 0, and that alone is able to, that the ASO that we use alone is able to increase the median life expectancy from 17 days to 29 days. But when we then couple that together with our base editing drug, we can now see the median survival increase pretty dramatically to 77 days. And actually, to be fair, the animals that die after this time point actually aren't dying of motor neuron disease. They don't sort of like slowly paralyze and decline in that way. They typically are just running around and then drop dead. And that's because this animal model, and this is specific to the animal, not human patients, has a cardiac phenotype that's associated with the disease as well. And as you can see here, this is an example of what these animals actually look like. So I think a video is probably more telling than a graph. This is at 200 days old. We have an animal that's treated with ASO and the base editor compared to one of its wild type litter mates. And as you can see, it's mobile and happily moving around. It's not entirely normal looking. You'll notice, among other things, it's missing a tail. But this also is something that we know we're not really worried about in case of human patients because humans don't have a tail, but also because the peripheral phenotypes, again, are ones that are related to the animal model and not to the human condition. So now that we had demonstrated this in this paradigm, and we're, as I mentioned, continuing to do the preclinical development of this drug, we wanted to go ahead and start working on an entirely different, also deterministic genetic disease, which is the first disease actually that was known to be associated with the gene. It was discovered back in the 80s, which is the Huntington gene. So the mutation in the Huntington gene is pretty much a very different form. It's part of a class of what are known as tandem repeat disorders, specifically trinucleotide repeat disorders. And these are caused by expansion of a repeated triplet of nucleotides. And so TNRs as a class include around 40 different disorders that are caused by somatic expansion of the repeat sequence. These include sequences like CAG, CAA, CTG, GAA, and CGG. And they frequently cause loss of motor coordination, and they're also very often fatal. There are many features that distinguish these different diseases, including the genes that they're located in, the location within the genes that they're located in as well, and the sequence itself. And the sequence itself is responsible for secondary structure that the DNA can form, and all complications that arise from that. And Huntington's disease specifically falls into a subcategory that we call polyglutamine or polyQ disorders, because CAG, when found in an exon, encodes for glutamine. However, both CAGs and CTG, which is its reverse complement, are found both in coding and non-coding regions of the genome, and they do similarly undergo these expansions that are associated with some form of disease. And then there's also Friedrich's ataxia, which is a disease that's caused by the expansion of these GAA nucleotides that are located in the first intron of the frataxin gene. So how do repeats end up causing these? Well, a common feature of all of these that I've described, even though they can differ quite drastically with their location, is that they are, in fact, all transcribed. And what happens during transcription is that an R loop can form where the newly synthesized RNA can hybridize with the template strand, leaving the non-template strand exposed. And because that sequence is highly repetitive, and in most cases, as you see, also very GC rich, this forms secondary structures that when the DNA is trying to hybridize together once again, can slip out. And these slip out structures then cause also slipping out on the opposing strand where you have these loops that come out. And these become a problem that DNA repair needs to deal with. And they also become more vulnerable to, for example, oxidative stress or cytosine deamination that also natively happens in the cell all the time. As a result of this, you get nicks, and these nicks then need to be resolved. And because the sequence is so repetitive, it becomes very difficult for DNA repair to know exactly where that repeat that it's trying to resolve was supposed to match. And so what happens a lot of the time is that it matches in an incorrect place, which then results in a change in length of the repeat. So it can either shorten, contract, or it can expand. And this is what we refer to as instability of the locus. And what we know is that the instability of these loci increases as the length of the repeat itself increases, most likely because, the longer it is, the more far away it is from the anchoring points of the side that aren't repetitive. And while it can both contract and expand, when repeats are on the shorter side, they tend to favor contraction more, whereas as they lengthen, they tend to favor expansion. And so you get this feedback loop where a long repeat then, when it's unstable, favors adding more repeats on. And so then it becomes again more unstable and is more likely to again acquire more, and so on and so on. And so that's how it has this essentially ends up being an out of control expansion of these repeat loci. And so what does it look like? It looks something like this. So here we can, for example, see a set of alleles. Okay. Oh, point me toward my computer. No, I think this works. Does this work? No. Well, anyway, you can see it over here. You can see a set of alleles basically that you can start with. So in this case, the median allele size is around 80 repeats long. And over time, if you look 6 months later, you can see that this peak has diverged, and the majority of alleles now have lengthened to get over 100 repeats. And there's also a subfraction that has become smaller at 76 repeats. And this is this instability phenomenon. And this happens in somatic cells. So patients in the germline, they're born with a certain length, and there are some somatic cells where this expansion can happen. One of the somatic cells in which this happens as well, if you can call it that, is germline cells. And so for a lot of these disorders during spermatogenesis, there actually also is an addition sometimes of one or more of these repeats. And what that can result in is that there might not have been a family history of, for example, Huntington's disease, but then suddenly de novo in a new generation, you can have now what will be hereditary Huntington's disease in that population. And this is a process that's called anticipation. As mentioned, if they are of a certain length, they will be stable. And so it should be noted that in all of these genes, the repeat sequences that I've showed you are present in the general population. We all have these repeats, but we have them up until a certain length that is considered stable. So in the case of Huntington's, having about 10 to 26 of these repeats is pretty normal, and that's most likely what most of us have. If you have about 27 to 35 of them, then you might be at increased risk of anticipation, where subsequent generations could gain the pathology. And then after that, if you are 36 to about 39 repeats, there is low risk, there's lower penetrance of the disease, and that probably relates to other polygenic risk factors, that some of which have begun to become elucidated. But 40 with Huntington's disease is a threshold. If you have 40 repeats or more, it is deterministic that you will get Huntington's disease. And the timing when you will get is dependent on that length as well. So as you can see, If you have a germline length, so born with about 40 repeats, you're likely to get Huntington's disease in about your 50s. But if you have 60 repeats, now you're more likely to get it in about your 20s. And in particular, in sort of that 40 to 45 range, every additional CAG that you have at germline length will bring the age of onset forward by over a year. So it's a pretty sensitive portion of this. And that's probably because of this fact that I was explaining, where more repeats beget more repeats. And so you are getting closer to that tipping point of where you sort of end up going more rapidly out of control. So one of the many curious aspects of this disease is the latency until the age of onset, because once the disease does manifest, the symptoms also tend to manifest more quickly. But it does seem to be true based on a battery of motor and cognitive tests that up until the point that patients get Huntington's disease, they are not subclinical. They do not have what seems to be some small degree of pathology that goes unnoticed, that slowly, linearly builds up, and then they have disease. They don't have it, and then it suddenly happens. So how is this possible, and why? To answer that, earlier this year, there was a really beautiful paper that came out from the Carroll lab where they were able to look at the striatum, so cells from the striatum neurons of deceased Huntington's patients, which is really, of course, as you can imagine, a pretty unique thing where they performed single cell on these brains, and they carefully studied the relationship between the transcriptome and the repeat length to try to see, how is it that Huntington's is actually progressing. And they were able to then subdivide the disease progression into different phases, A through E. In phase A, it's the slow expansion phase. And this phase lasts several decades. This is where patients do not at all have any symptoms. And apparently this is up to about 80 repeats long. Then phase B, after this tipping point of 80 repeats has been reached in some somatic cells, this expansion rapidly accelerates. And then once you hit 150 repeats, now these cells suddenly begin to have over 500 or more genes that become dysregulated. At this point, the cell is losing its cell identity. And ultimately, as this exacerbates, they get de-repression of certain key developmental and senescence and apoptosis markers. And then after that, very quickly, the neuron dies. What is also really important to note here, however, is that it seems to be that there may well be a reasonable therapeutic window for treatment of patients based on their analysis. Not only if you can catch neurons before they hit this 150 mark, and so before patients begin to have any symptoms of Huntington's disease where many neurons can be lost, but even when neurons are beginning to be lost and patients are starting to suffer from Huntington's disease, the majority of the neurons in their brain actually have not yet met this point. So even at the point that about 25% of these neurons are lost, 90% of the neurons have still not actually hit this 150 point, which means that if you can find a way to intervene in this process of this expansion that ultimately leads to disease, you would have an opportunity to rescue the bulk of neurons and treat patients. And if you wanted to resolve it altogether based on sort of their modeling, if you had someone born with about 40 repeats, which is most people, then if you would be able to treat them before they're pretty much in their 40s, you might be able to hold off Huntington's disease at all. So what would be a way of being able to intervene in this expansion process? There was a GWAS study that came out in 2019 that looked at genetic associations with Huntington's disease. And the major finding from that is actually that the sequence itself is extremely important in this expansion process. So in most people, I believe also including most Huntington's patients, the CAG repeat, the poly-Q region, is not just CAGs. There is a penultimate CAA that happens, which also includes Q. So the poly-Q length is actually two codons longer than the CAG repeat stretch is. And that is considered sort of this gray normal line that you can see here in the middle that predicts your age of onset based on length. However, very often, and more so in Huntington's population than in the general population, that penultimate CAA is lost. And when it is lost, which is the red line, you can see that the age of onset is approaches much more quickly at the same poly-Q length than it does if you did have that one CAA interruption. And then in some cases, that CAA-CAG is actually duplicated, and there the age of onset is much further. So having just a single or multiple of these CAA silent codon changes is able to have a pretty profound impact in the age of onset of this disease. And if you look overall across the population, patients that have pure CAG repeats versus poly-Qs of a similar length, but with a single CAA interruption, that delays the onset of Huntington's disease by 25 years, which is obviously quite a profound difference. And from what we know, this isn't unique to Huntington's disease. So Friedrich's ataxia, as I mentioned, is another disorder that is caused by GAA expansion, so a different sequence altogether. There isn't as large scale of study as was done for Huntington's disease, but here it is also apparent that having interruptions in the GAA repeat tract that's found in the intron of GGA, GAG, GGG is able to also very significantly, by several decades in some cases, stave off this onset of the disease, most likely, again, due to the repeat expansion. And this, to a certain degree, had been known from prior work on bacterial cells, yeast, where people had inserted these sorts of sequences to study the process of repeat expansion, that these interruptions really are very important in determining the frequency of repeat expansions and the rate of it. So why is this important? We have a technology that allows us to make single nucleotide changes in the genome. And so we set out to develop two different strategies, one targeting on the opposing strand of CAG, so CTG, a genome editing strategy that's able to convert CAGs into the synonymous CAA codons. And then on the GAA side, one that's also able to insert A to G changes in the GAA repeats in the intron to hopefully have a stabilizing effect on these repeats. So it is a bit of an unusual strategy for genome editing, which is typically the goal is to be as precise as possible and target only one genomic location. Here we had a different goal, and here we designed our guide RNA in a way to essentially be somewhat non-specific to the entire repeat region. Our thinking was that it would allow it to somewhat sporadically intersperse interruptions across the length of the repeat tract. And with longer repeat tracts, it would allow you to insert more of these stabilizing mutations. So we developed a strategy that would then, as mentioned, use cytosine deamination to convert CAGs into CAAs. And we looked at a bunch of different strategies to do so with the available deaminases at the time. And we were really happy to find that a great number of them were able to edit this locus with high efficiency. So this is in a screen in HEC293 T cells that have only 17 CAGs, so relatively short, so it's a normal number. And I want to draw your focus initially to the ones that are on this side of the graph. And you can see that the editing itself is quite high, but we also have these other byproducts that are not as desirable, that you can see in particular these stop codons. So where are these stop codons coming from? As mentioned before, the reason that cytosine deamination happens in the portion that you're targeting your guide to is because the guide RNA binds the other strand and it makes available the single-stranded DNA on the opposing strand. However, we have seen in the past from high-throughput characterizations of these tools that you can sometimes see editing happening outside on the PAM distal end of the guide RNA sequence that you're targeting. And in particular, this is also noticeable on the opposing strand that you're getting some of these deamination events. And this is likely due to breathing of the DNA as you're having this binding event here. And this is typically a very rare thing that happens only with some cytosine base editors, but most likely because we're having so many consecutive binding events happening, you might have more of that. And so we actually see some editing of the opposing strand here. And that, in this case, would result in TAA or TAG stop codon formation. Now, this is not something that we want. We think, at least. We probably don't want to knock out the Huntington gene. So we made a bunch of architectural changes to alter the frequency of these still relatively rare outcomes, and also somewhat increase efficiency. And ultimately, we picked a strategy, which is this 32 NLS strategy, to move forward with. And then we took this strategy and we wanted to apply it in the context of longer repeats, because we don't know how it will behave. And just because it does a certain thing at 17 repeats, we don't know what it will do at longer repeats. And so we looked at patient fibroblasts that have different allele lengths, which can be seen here, shown at the bottom. So there's typically a normal allele in Huntington's patients and a longer allele, as can be seen here. So for example, this patient has one allele with 17 repeats. But we can see across the board that we actually get high editing at both alleles, both the long and the short one, in each of these cell lines. And there always seems to be, in fact, a little bit of higher editing from what we've seen in the longer allele and shorter one, which could make sense in the sense that, it's more of a magnet, essentially, for multiple binding events as it is longer. One slight caveat to, in particular, this last one here that has a very long repeat of 180, we do this analysis by short read Illumina sequencing, and we're actually only able to cover about half of the repeat tract for that 180 allele. So we can cover the other alleles because they're within what we can sequence, which is about 90 of them, but we can't fully cover the 180. So the actual editing at that allele might in fact be higher than what is shown here. Another thing that we were quite happy to find is that the incidence of stop codons we actually didn't really see them here. There was some minor fraction of indels, I believe, but nothing was over sort of the untreated condition because these loci are difficult to sequence, so it probably wasn't even a real finding to begin with. So that wasn't a concern anymore. Then the, I think, more interesting finding is what does this do to the repeat tract? So here we can see essentially at baseline the two alleles. So we have the allele histogram for the short allele over here and for the longer allele over here for this 18180 cell line. This was the only cell line with the longest repeats that we could even see expansion happening in culture, which, kind of makes sense because we can't culture these primary cells for so long to see this process happening. So we monitored these cells either untreated in blue, which is, I don't know if you can see very clearly, but it's hiding behind this purple, or with the cytosine base editor treated samples. And then we cultured this over 5 passages for about 30 days. And what you can see here is that the untreated expands and gets larger in almost all of the baseline longer alleles. And our CBE treated stays pretty much stagnant, and in fact drops in size a little bit. So this one has increased by, I think, about 5 or 6 repeats, and this one's actually dropped by about 5 or 6 repeats from our starting point. So we've, in fact, demonstrated now that experimentally, if you go in to long repeats and you make these edits, you do, in fact, confer the stability that has been found previously by association in the human population. So then, as mentioned, the guide RNA that we're using is not specific. It targets the repeats. There are more of these repeats in the genome that don't just belong to the Huntington gene. Importantly, there are actually about a dozen or so different trinucleotide repeat disorders that are caused by CAG-CTG expansion. And of course, our genome editing strategy targets each of these essentially indiscriminately. What's really interesting about this is that, if you could envision in the future, you would have such an approach that you may use in the clinic. If you validate a drug for any one of these targets, that means that that drug would also then work for any of these other trinucleotide repeat expansion disorders. However, aside from ones that are associated with disease, and of course, in any one patient, only one of them will be associated with disease, the other ones are still there. There are very, very many other loci throughout the genome that also have around 8 or so, 7 or 8 of these repeats that will also be targeted by the strategy. So to sort of get at how many these are, where are they, what are they, we performed Circle-seq initially. So Circle-seq is a biochemical assay where you use a nuclease form of the Cas protein with the guide RNA to nominate loci where the base editor might land in a cell condition to induce base editing. And we found well over 5,000 of these loci throughout the genome, of which about 10% were a perfect match. So they also just had pure of the CAG repeat. So they're not even off targets, they're alternative targets of our guide RNA. And then subclasses that had increasing number of mismatches, which we know are likely in the base editing context in cells to actually reduce the potential for editing events there. And then we wanted to look further into what these were. And so we were able to subdivide that out of these 5,700 loci that are potential targets. About 17% of them code for proteins. And there are, of the protein coding off targets, about 15% of them that are a perfect match for our CTG. The CTG or CAGs very often encodes for glutamine, but sometimes it's in a slightly different frame where it can also encode for other amino acids. Of the 4 different amino acids that we found that it encoded, 3 of them are synonymous changes, glutamine, leucine, and cysteine, but serines will be converted into asparagine, so that would be a missense mutation. So we were interested to find of the roughly 1,000, in particular the 1,000 perfect match protein coding loci, what those are, but also the other off targets, how frequently they're actually occurring and what they're doing. Now, usually when you do Circle-seq and for this sort of validation, you pick the top or maybe the entire list of off targets to look at in the genome to see whether or not there's editing, but there's thousands of loci here. And so that's not really something we can do in an arrayed, single locus amplification fashion, which is what we typically do. So we sought instead to do whole genome sequencing in this case, at very high coverage, 160x, to be able to see after we treat cells with our genome editing tool, what is our likelihood of actually getting an editing event there. Even at 160x, it is pretty difficult to get good quality calling of a single nucleotide change that happens, in particular in repeat regions that are very difficult to get through and are more likely to induce errors. But if we did this nevertheless, and if we look at any loci that have over 0.5% over background of a single nucleotide change, there's about 48% of Circle-seq nominated loci that we would call as an edit, which is about 2,743 loci. We did find, as anticipated, that the frequency of edits decreased steadily with the number of mismatches. As soon as you have, essentially over 2 mismatches, you don't really get editing at these loci, which is pretty much to be expected. We demonstrate this here in a table where we basically subdivide where the mismatches fall. So it had been known from prior work and was reproduced here that mismatches in the guide RNA that are more proximal to the PAM, particularly in the seed region that are shown here in A and B, but the closer, the worse it is, that those have a greater impact on the binding ability of Cas9. So if you have mutations here, that's worse than if you have mutations in the tail end. And that's what we can see here. These are ones that have no mutations at all in the entirety of the guide RNA. Here you have ones where you have no mutations in A, but you have only mutation in the C region, etc. Then we wanted to some degree validate these editing events by doing single locus amplification. We find that for the most part, our whole genome sequencing analysis is pretty accurate at calling it, if overestimating to a certain degree due to our sort of liberal calling methods, what the editing frequency there is. But so ultimately, we were most interested in what we thought might be mutations of clinical significance. So we found that indeed the overwhelming majority of edits were non-coding, intergenic, or in other regulatory regions, or inducing silent mutations. And there was a very small subset where there were, relatively compared to what we started with, where there were potential changes. So if we looked at whole genome sequencing loci that had over 5% editing to call it somewhat more accurately, we find that over 1,200 sites show editing, including 243 genes. And of those, 91 genes in total have mismatch mutations. So then we wanted to ask, what are these genes and how important are they? So we compared them with the cancer depth map to see if any of these genes are cell essential. And we also looked whether or not these genes are expressed in neurons because those are the ones that we're delivering the editing to. And then we also looked at alpha missense to see if we're making these non-synonymous changes, what is the likelihood that that will have an effect on protein folding or protein function. And ultimately, we found that there were 4 genes that were essential that were targeted, one of which has a mutation that is predicted to result in a change in protein folding or function. And we only actually throughout the entire genome found with our strategy that we'd optimized to induce a stop codon, which was also really nice. But that stop codon was in a gene that is neither essential nor expressed in neurons. So it's probably not relevant as it's not transcribed in the cells we're targeting. So in sum, the base editing approach has quite a lot of off targets that do happen, some of which may be of clinical significance. So if this is something that we would ultimately want to pursue more for a therapeutic angle, we would need special attention on those sites in the future. So ultimately, we then wanted to take this same strategy and see what happens when we put this into an animal system. So not just cells in culture, but in an actual organism. And so we did the same form of delivery where we used AAV9 to inject this base editing strategy. We do this alongside a small dose, a tenth of a dose of GFP, which is basically just a tracer control for transduction efficiency. And we can see that when we sort out these nuclei and editing pieces over weeks, but at 24 weeks, we have around 80% editing of targeted nuclei. And what does this do to the repeat alleles? So we monitored various cell types over a time course, so 12 weeks, 24 weeks, and we looked at cells both that expand and don't expand. We were really excited to find that similar to what we found in our cell culture conditions, when we look at the striatum, which is the disease tissue, and that also has the most severe expansion phenotype in the brain, we can see in pink relative to gray a reduction in these the longer hump of expanded alleles. And in fact, we're also gaining these contractions here shown here in pink that you don't have in the control condition. So it's probably easier to see than in that histogram in a plot like this. This is an analysis called the instability index that, as the name would suggest, measures the instability of alleles. And as you can see here at both 12 weeks and then more pronounced at 24 weeks, in cells that have, in animals that have received the base editor, we can see a significant drop in the instability index compared to untreated animals. So this is very exciting. When we were working on this paper, another study came out where they actually generated animal models that had either just the CAG repeat, as is the classical version to have in that mouse, but they also made versions of that same allele that had CAGs interspersed. I think it was every third or so. CAG was a CAA, and they demonstrated there also that freezes completely the expansion of repeats. And here we were able to show for the first time that inducing these into what are already long alleles in an animal system where these alleles expand, you are actually also able to do that same thing where you freeze these. I will also note that these data also have to be qualified by the fact that we didn't have a way of sorting out only those cells that had been edited. So this is actually looking at all cells that we were able to isolate. And as we know, there's only a subfraction of cells. If you look at the bulk, basically, that's here about, there's only maybe about 35% of neurons that even have the opportunity to be edited in the first place. And so this is, again, most likely an underrepresentation of the actual rescue that the approach of inducing edits actually has on alleles. And it's more likely that the alleles that have edits are likely to be frozen. So then we want to do the same strategy for Friedrich's ataxia. So I think you all get the the idea here. So we have a guide RNA that targets the GAA repeats, and we want to turn some of those GAAs into having some A to G interruptions. We try to several different Cas proteins to target in different frames to induce these sorts of edits. And we were able to do so with pretty high efficiency that we were quite happy with. One thing I'll point out here is that we have the purple bars, which are, which is actually, I think I put the wrong graph up here. I wanted to show the difference between nickase-mediated editing, which is the classical base editor, and this dead Cas9, but this still suffices. Ultimately, we went for the AB8E deaminase, but what we did is we actually paired this with a dead Cas9. So as mentioned, typically you have a nickase that nicks the opposing strand to bias editing toward incorporating the edit that you want. But we decided in this case, because the editor was in fact so efficient, that we might not need this. And we made it into a dead Cas9, meaning that there was no nickase activity and there's only the deamination. So you're going to have fewer complete conversions, but the editing is enough that we thought that was fine. Why did we decide to take this approach? Similar to CAG-CTGs, that there are many of in the genome, there are orders of magnitude more GAAs. The GAA repeat in this location, as well as across the genome, are in ALU elements for the most part. And these are dormant retrotransposons that we have interspersed all throughout our genome in many, many different locations. For the most part, they perform regulatory functions and introns and ITRs. But just due to the sheer number of them, we felt like maybe having nicks all over the genome in so many locations might not be as favorable. And since it wasn't really strictly necessary, we decided to omit that nicking feature. Then we took our best strategy, and this was done in Huntington 3 cells that only have 9 GAAs at this locus, which is not a lot. So we wanted to have transgenic cell lines where we had more of these repeats. So we derived mouse embryonic stem cell lines that had either 30 or 60 GAA repeats, which is over the pathogenic threshold. And we ultimately chose our top favorite strategy, which is NRCH AB8E editor that is able to still induce edits at a relatively high frequency. And then we also did the same thing where we targeted patient cells. Patients with Friedreich's ataxia have a much greater number of repeat expansions. The expansion process goes into the thousands for patients that have that. So it's a slightly different problem than you have with CAG repeats, where you're already very much pathogenic if you're at the 80 range. So these patient cells had short allele 300, 330, and 380, or 541 and 420 alleles. And also, actually, an important thing to note is that while Huntington's disease is a dominant disease where one allele is pathogenic, in the case of Friedreich's ataxia, it's actually recessive. And so patients will have 2 alleles that are affected. So here we performed editing again and measured the editing that we got at these repeat loci. Again, here we're stuck with this limitation of Illumina sequencing, where we can only sequence about 300 base pairs, which covers for us about 380 of these GAAs. And as you can imagine, even in our shortest of these alleles, that's 330 repeats long, that's a very small portion of it. Now, we sequenced it both from one end and the other end, and we have essentially the same data, suggesting that the editing, as expected, is happening uniformly across the repeat, which is also what we see in any of the shorter, long repeat alleles. But because this is such an underestimation, we developed this extrapolation model to say, okay, if this is the amount of editing that we have here, what does that mean? Can we estimate how much editing we'd have across the rest of the allele and determine what the editing probably actually is at these loci? And so we estimate that our editing at these alleles is actually around about 80%. Yes. Oh, okay. Yeah, I'm almost done, actually. I think I only, yeah. So then one other valuable thing about Friedreich's ataxia is that we actually know the cause of the disease is that when these repeats get so long they silence the genomic locus, and there is reduced expression of the FXN gene. So we want to see, does our editing actually affect this loss in expression? And we were not necessarily expecting that it would per se, but it did, which was quite a good finding. So in untreated short normal fibroblasts, the level of expression we basically normalize to 1. And then if we compare patient fibroblasts with the long repeats, either untreated or treated with an unrelated guide RNA, you have only about half of the expression, and you rescue about half of that loss by doing this base editing interruption. I guess I'll breeze past the fact that we also did whole genome sequencing and circle-seq here, as mentioned, an order of magnitude more hits, but actually a way smaller subfraction that are coding, because again, these repeats mainly arise from these allo elements that are regulatory sequences. And we did not find any stop codons, and for the most part, our base editing strategy just mimics the natural variation of inducing these A to G changes in repeat tracts. There are a very small number that do warrant further study because they are potentially in regions that might have a negative effect. And then we did this in animal models, so just take you through that quickly as well. Both in animals with 300 repeats or 800 repeats, our estimated editing is around 60% of alleles. And this has a profound impact on the instability index as well. If we look here at the cortex, where we can have significant expansion, our edited cells have much more stable alleles. And this is even more striking when you compare the longer 800 mouse relative to the 300 mouse. So I will leave it at that. Thank you very much for listening. I want to give a huge, massive shout out to co-lead author on the paper, Shannon Tuzik, who's a brilliant now postdoc in Paola Arlotta's lab. She was a graduate student in David's lab where I was doing my postdoc, and that's where all of this work was done. So also major shout out, of course, to David, who allowed us to do all this incredible work. This was also done in collaboration with Ricardo Muro-Pinto's lab. He runs a lab over at MGH and is an expert in both of these and other repeat expansion disorders. Guangping Gao's group, who was able to help us with virus. Arthur Burgess, who also collaborated on the SMA project, and Kat Lutz over at JAX. Everyone from my lab, some of whom are shown here, who have also helped, some of whom helped take this paper over the finish line. And of course, our funding sources that NIH who's able to help support all this really important work. So thank you very much.